This Week in Virology, the podcast about viruses, the kind that make you sick. From Microbe TV, this is TWiV, This Week in Virology, episode 1197, recorded on February 28th, 2025. I'm Vincent Racaniello, and you're listening to the podcast all about viruses. Joining me today from Austin, Texas, Rich Condit.
Oh, man, I thought you were going to do somebody else first, and I don't have the weather up yet. Oh, here it is. Okay. Hi, everybody.
I randomly pick.
Yeah, you were flipping a coin there or something like that. So it is 77 degrees and sunny enough to get just a little hint of Dixon over my shoulder right here. Okay. Maybe it'll clear up more and we'll get more of them. So it's good. All good.
It is 9 degrees Celsius here in Chelsea. It's got
Vincent is seized up on us.
A little bit of sun. But
Everybody's
Saturday it's going down below freezing again. Jeez. Also joining us from Western Massachusetts, Alan Dove.
Good to be here. It is currently 45 Fahrenheit, 7°C. The ice and snow and more ice is mostly melted, but in the process of all those storms, I noticed my my wire ham radio antenna had blown down out of the trees, so I gotta launch that back up.
It was in the trees. That's where you keep it?
Yeah, that's where we've got these huge, 75-foot tall oak and maple trees. Great antenna supports. So it's
how do you get up there?
Oh gosh, I have the coolest thing. I call it an antenna launcher. Other people would call it a potato gun. But you use a weighted tennis ball that I've got a fishing line attached to, and there's a fishing reel on it. It looks totally like Rube Goldberg. And then I pump it up to about 20 psi with a bicycle pump. It's made out of PVC. This whole thing is made out of PVC pipe with some irrigation fittings. And then I squeeze the valve on the side, and it goes boonk, and the tennis ball will fly easily 100 feet up and carry a fishing line over whatever branch I've aimed for.
Oh, cool. Also joining us from Madison, New Jersey, Breanne Barker.
Hi, great to see everyone. It is 48 Fahrenheit, so also 9°C here, and sunny. And I have no exciting potato gun stories.
Potato guns? If you enjoy these conversations about science, and this is where you can get some science. There are other places, but we've been doing this for a while, and we want to give you different sciences on different podcasts. This one is about viruses. If you want to
and also, radio and ballistics and weather.
Yeah, potato guns. We'd love your support. We don't do ads. We just rely on your donations because we're a nonprofit, and your donations are U.S. federal tax deductible. Although who knows, tomorrow that could change. Everything is changing. So you can go to microbe.tv/contribute, different ways that you can help us. ASV 2025 is going to be at McGill University in Montreal, July 14th through the 17th, asv.org/ASV 2025. Early bird registration ends May 12th. They got a little bit of time.
Get your birds registered soon.
And current members get a discount on their registration rates. And you might want to look for housing. It's a good time now. If you go to the ASV website, you will find information about housing on travel and accommodations. There you go. We have news. We have a lot of news today. Ann Palmenberg sadly has died. Ann Palmenberg was a virologist at the University of Wisconsin-Madison. She worked on coronaviruses, so I knew her very well because that's what I worked on, and I saw her a lot. She's been on TWiV a couple of times, I guess. Maybe, well, I don't remember. I know I interviewed her in Madison at least once, and then she gave a talk at a symposium for Kostya Chumakov, which took place a couple of years ago, and I'll put a link to that video in the show notes. It's pretty funny stuff going on there. But I liked Ann a lot. She was a major force in virology. She was a big supporter of Women in Science and the American Society for Microbiology. It's been at Wisconsin pretty much most of her career, and mostly worked on coronaviruses and proteases and their role in the reproduction cycle. Big football fan, always had season tickets to the Wisconsin, what are they, Badgers?
I think
They
they're
are
Badgers.
The football team. Big bloody stadium there. In fact, I used to stay at this hotel, which was right across the street from the stadium. She always told me stories about season tickets. She would say she would get a new recruit, and she'd go, "You got to put your name on the list for season tickets now, or you're never going to get them." Season tickets, and she made me this. Here, let me see if I can grab it. So, many years ago when I visited her, she gave me this plastic model of polio virus, okay? It's made with these plastic subunits. They're old and they're painted, but it was cool at the time, a long time ago. And then I visited her once, and she had a beaded genome of rhinovirus, okay? So the whole viral genome, and each bead is a different base, right? And I looked at it and I said, "Oh boy, I'd love one of those for polio." She says, "I'll make you one." So like 4 years later, I got it in the mail.
7,441 beads later.
Yes. She said, "I did it during the World Cup. It was so boring." So you could see, maybe you could see here, this is all red. That's A. It's a poly A tail.
Right.
And I've stuffed it into her original polio virus that she gave me.
Nice.
And the DNA is streaming out. The cool thing about this is, oh, look, we have VPG here even. So this is the 5' end. There's a protein link to it. See?
Mm-hmm.
There it is. Each gene segment can be uncoupled from the next. She put a magnet in between each one.
Wow.
So I could take the 5' UTR off. I could take whatever, Very cool. So I love this.
Boy, that is a labor. That is a
That's
labor
awesome.
of love. That is just
Yeah,
awesome.
every base in sequence, right? 4 bases there. Lovely.
Well,
Here's a
I was
magnet.
going to say of Ann that she was just tireless. She was just a bundle of energy, and that proves it right there.
Yes.
Because
Yeah, she was very energetic. Yes.
that genome thing. That's extra credit, I think.
Yeah.
So Ann gets lots of extra credit, and she was amazing. She's going to be
And an
missed.
analogy just occurred to me between viral genomes and a rosary. It's like,
Yes.
and then
Yes.
So
Where-
she,
So those are the things that you're learning this week about viral genomes. This week I was writing some sample sequence on the board that was going to be viral genome. And I learned that with the RNA bases, you can spell guac, which I did by accident.
Yep. Yes, you can. There will be a
But that's
celebration.
extra.
Yeah. Celebration of her life in the spring, so we'll keep you posted. All right. I'll put a link to the Chumakov talk also in the show notes. Okay, Alan, you put the next one in, or no? I did.
The deadly illness one in Congo, I did not put in. I had seen this,
Okay.
but
All right, so I put in a Times link, which I
Yeah.
can't even read. But,
it loaded
there's a
for me.
deadly illness going on the Congo again because there was just one last year, which turned out to be malaria, a severe form of malaria. This one, I don't know how many people have died, a lot. And the people who die within 2 days, which is incredible to me. I think there were over 100 deaths, over 100 infections.
431 reported cases, 53 known deaths.
Very quick.
Half the deaths happened within 48 hours of symptom onset. So this is something fast. And apparently it happened right after 3 kids had eaten a bat.
Yeah, I've noticed that part.
And
stood out to me
they
as well.
were index cases. That was in January.
They did tests for Ebola and Marburg, and they're negative.
Yeah.
Over half of them had malaria, which is not uncommon in this area. But we don't, as of this writing, we don't know what it is.
Yeah.
Okay.
And this is in the northwest of the country. The eastern part of the country is engulfed in civil war and is even more of a mess.
Okay. Did you put in the
I
Trump
picked
shenanigans?
the next 2.
Okay, go ahead.
So, first of all, you've probably seen the news about the Zitter CEO, who's now been mucking around with the federal government, having no idea what he's doing and just turning things off. And he shut off USAID because, gee, who needs it? It turns out that's going to be making mpox great again. So if you look at the official statistics on mpox in here we are in DRC again, aren't we?
Yes.
Yeah. So there was an mpox outbreak going on in DRC, and then the numbers appeared to get better. Number of cases dropped. Turns out that is just an illusion, because the number of cases dropped because nobody's counting them anymore because those people got sacked.
Nice.
And the funding got withdrawn, and so, it's spreading more and more, and now we're getting some reliable reports on the ground that this is turning into a bigger and bigger outbreak. So that's going to be huge. And then the next one.
I just want to say, no one is safe until everyone is safe.
Yeah.
Mm-hmm.
Yep. And, if you say, mpox, it's some blisters and it's not that big a deal. Next up, we have ebolavirus, where Musk accidentally cut the funding to ebolavirus prevention efforts and response efforts at USAID and claimed, oh, no, we did that by accident, and then we restored them immediately and everything's fine. It may or may not be true that it was done by accident. It is not true that they restored them immediately, and it is absolutely not true that everything is fine. So this is one of these things where you can't just flip these switches off and then back on and expect the lights to come on again. And so, we have mucked up our Ebola response, and the U.S. is the major funder of this sort of thing. And so this is in, what do we got going on? Is this in West Africa again? So outbreak monitoring is all mucked up now. The emergency response branch of USAID, because USAID has been shut down, that's gone.
Mm-hmm.
And I don't have, Uganda is having an outbreak right now, and we're dropping the ball.
So we live in a global community, like it or not.
Yes.
There was a time, I think, when you could be an isolationist and get away with it. But I don't think that's the case anymore.
That time
No,
ended
not at
several
all.
centuries ago.
Yeah, and so everything is interconnected, and you can't just shut off something like USAID and think that it's a surgical act. Everything is interconnected, so you shut off one of these switches, and it has a ripple effect that is just phenomenal.
Yeah.
So
This is because the people shutting off have no clue how things work.
Right.
They don't have any idea.
No idea. They're really ignorant.
Yeah, I drew the analogy before to a new user of Linux who runs the top command or the system monitor and says, "Oh, what are all these daemon things? I'm just going to stop those."
Yeah.
And then the system shuts down and they don't know why. That's what Musk is doing. He doesn't have any idea what these agencies really do. And as I commented before the show, this, even if you're like a diehard America firster and we should be in charge of the world, but also looking out for ourselves, this is complete lunacy. Because what's going to happen is there'll be this huge power vacuum, and China and Europe will rush in to fill it. And when we do recover our senses and go back, our previous role in the world will no longer be available.
So I don't know if anyone who voted for this administration is listening, but this is what's happening.
Yep.
And it's not good. There's no justification for this. Well, they're justifying it for money, right? They want to get money to more
Which is stupid.
wealthy
It
people.
doesn't even work.
So,
Yeah,
but
USAID I think is far less than 1% of the federal budget. So that doesn't seem like the great place to go for your
piddlingly
savings.
inexpensive things that they're cutting, and of course they're not going to touch DoD. So yeah.
every news item we have has to do with this silliness.
Stupidity, yes.
We have as you know, there's a measles outbreak in West Texas. We talked about this last week. Case count is now over 146, and there's been a death in a school-age child, yesterday. And this is in a community of Mennonites who choose not to immunize. And one wonders how you feel if your child dies because you didn't immunize them. I think you must feel terrible, right?
Yeah.
Because it's totally preventable. So this is unfortunate. And, there are pockets of measles susceptible people throughout the country like this. And so all this virus has to do is get into one of them. We have some measles in New Jersey. I'll wager it's just a matter of time before it comes here to New York City. All of this due to anti-vaccine sentiment, either anti-religious exemptions, medical exemptions, or parents who believe RFK and Andrew Wakefield instead of scientists. I just don't get it. It is a world that I do not comprehend. And RFK, by the way, seems to not care about this at all.
Yeah, some of
He
his
says-
recent
says,
statements have been not great.
Deaths happen, outbreaks happen, it's no big deal. Well, it is a big deal if you can totally prevent them with a vaccine.
Yes, and they, people don't get measles and die of measles all the time. The last measles death in the United States was 2015.
The Times reports that the FDA has canceled its meeting of VRPBAC. What does that stand for?
The people who pick the strains of flu vaccine.
Yes.
That's right. People who pick, because as you'll see in my pick, the WHO has issued the strains for the next season. And then usually this committee meets and says, okay, and then the CDC recommends it. But they canceled this meeting. Why? Who knows? They don't tell you why they do any of this stuff. And this is just going to delay strain selection and could impact release of flu vaccines this fall.
Well,
It's
it's
just
because
idiocy.
they're canceling everything with health agencies. It's part of that flipping off the switches thing.
Yeah, so they don't know what is urgent and less urgent because they don't know anything. They don't know anything about science or vaccines or anything. Okay, they're just ignorant. And if there are scientists there, you're ignorant for being there. Okay, you should be getting out, not the people who are being fired. Of course.
VRBAPAC is the Vaccines and Related Biological Products Advisory Committee. And just to remind the listeners, flu changes all the time. And so there is a global network of influenza surveillance that every year tries to anticipate what the dangerous strains, what the predominant strains are going to be in the coming year. And there's a timeline to that because they have to because it takes 6 months from the time they pick the strains to get up to speed with manufacture of the vaccine. And that's what this committee meeting is about. It's about making the decision in a fully transparent fashion so that anybody who has a differing opinion or questions or whatever, it's public and they can do that. So that's not going to happen.
And this is a massive blow. Okay, yeah, I mean, public health implications are a big problem here. But I just want to point out, again, for any Republicans who are actually listening to this show, this is a massive blow to business, to a multi-billion-dollar business in the pharma industry, and to some companies in particular. I'm thinking of like Sanofi, that produces a huge number of flu shots. They have factories that they need to set up now with production for the flu vaccine. And what are they supposed to do?
This, I believe, is a committee that Paul Offit is on.
Yes.
Yeah.
Well, he's on ACIP.
He's on ACIP. Yeah.
So what did Daniel say? Okay, so Daniel said on clinical updates, so this is recorded, he said he had inside information. They're looking for a bigger room because they want the public to come to the meeting. Do you know what a mess that's going to be?
That's the point.
I'll go.
Yeah, they're going to be for you, every 100 anti-vax people who are going to scream and yell. You know, the CROI meeting, Conference on Retroviruses and Other Opportunistic Infections, that has traditionally always allowed the public in. It's a mess. They're disruptive. So I'm not sure why they have to do that, but to try and sway information away from the scientists. Okay. Anyway, the last news item is a Times article that came out yesterday. U.S. terminates funding for polio, HIV, malaria, and nutrition programs around the world. So emails went out from the State Department to all these places around the world, 5,800 projects that we support. These are part of USAID. And they include HIV treatment programs, polio immunization programs, a contract for bed nets, malaria tests and treatments. It goes on. It goes on. A project to trace contacts of people with Ebola in Uganda, to help bury people who died of the virus, medical supplies in Kenya, shelters who took care of 33,000 women who were victims of rape and domestic violence in South Africa, a project in DRC that operates the only source of water. All of this canceled. The switch off.
Yeah, I know that with the HIV programs in South Africa, in the past, and thinking about HIV in South Africa, there is some things in South African politics where people have talked about a past president and how some of his actions potentially led to a lot more cases there. In some cases, they say that there are something like 300,000 more cases because of his policies, and it is estimated that these cuts to HIV programs will actually lead to even more cases than that. So they're estimated to lead to 500,000 more cases of HIV just in South Africa.
No sane person would do this.
So on the theme of no one's safe until we're all safe, there are those who, if there is a rationale to this, it is that, we should be spending our money on our own people, and what are we doing supporting all of these programs that really, in other countries that don't necessarily affect us, which is not true. The example that I saw that somebody, it wasn't Paul Offit, somebody recently on the news gave, was the smallpox eradication campaign, okay? Where, interestingly, Russia and the U.S. were major collaborators in the final smallpox eradication campaign, which had to do with going to many third world countries where smallpox was still cooking. And launching the eradication campaign to make sure that there was no smallpox anywhere so that it wouldn't come back and bite us. Okay, so that's and now smallpox is gone. All right, had we not done that, it would be a totally different story. And that's, I think, a really good, clear example of how it is that public health in other countries impacts us locally. It is a global community, dudes. Okay, we have to pay attention.
And there are additional levels to this too. I mean, if you ask yourself, why did the U.S. and the Soviet Union, at the height of the Cold War, collaborate on this? The answer is because if the Russians did it and we didn't, they would have gotten a lot more influence in those third world countries, which are strategically important. So the hard-headed folks in places like the Pentagon are thinking along those lines, and yeah, public health is nice, but we also have to kind of keep up other things, and that's all part of the package.
We need friends.
We need friends, yes.
I think that the U.S. has always been able to support other countries, and it should.
Yeah.
We're not lacking for anything here.
Yeah, and there's the ethical aspect that to those whom much is given, much is expected.
I mean, you give a tax cut to billionaires, that's a lot of money wasted, in my opinion.
Yep.
And so this is not the place to take it. Ridiculous. Totally ridiculous. Okay. However, what is not ridiculous is science, and we're going to talk about science today.
Believe it or not.
And I think science papers will continue to be published for the foreseeable future, and we'll continue to report them. first paper is in mBio. Summaries. Alan, are you prepared to do a summary?
Oh gosh, I forgot. I didn't.
That's okay. Don't worry about it. We'll just do the paper.
We can
So
improv.
mBio, clade. There's going to be a quiz here, so remember. Clade 2.3.4.4b H5N1 neuraminidase has a long stalk, which is in contrast to most highly pathogenic H5N1 viruses circulating between 2002 and 2020. The B, Daniel taught me this, bovine. That tells you that's the lineage that's going in cows.
And let me just rewrite this title, which I did in my head when I saw it, which will also serve as a little bit of a summary. In contrast to most highly pathogenic H5N1 viruses circulating between 2002 and 2020, clade 2344b H5N1 neuraminidase has a long stalk.
It's very good. Very good.
I eliminated 2 words, and I got the punchline at the end.
This generally speaks to the question that I had the other day, which is we have now these H5N1 influenza strains that are going into cattle. Some show up in people, some in birds. And what are the genetics of these things? Are there genetic changes that account for differences in transmissibility, differences in host range? And this tracks one genetic characteristic, which is the length of the neuraminidase stalk, and tracks it and speculates on the importance of what they find.
Yeah. So the authors are Eniko Herman and Florian Kramer, and they have adopted the incredibly annoying Nature habit to see affiliations go to page 6, which we do.
Disgusting.
And they're Medical University of Vienna and the Icahn School of Medicine at Mount Sinai.
So I picked this paper because I'm not so sure the stalk is really important. I mean, there's some circumstantial evidence. It's just not clear. But it's got a nice summary of H5 genetics and what's going on. So I thought for that it would be cool. Okay, so this clade, the B clade, 2344b. So first of all, H5N1 emerged in 1996. Highly pathogenic H5N1 emerged in 1996, caused an outbreak in Guangdong in geese.
And it's highly pathogenic to birds.
Right, to birds.
Yes.
Right. And then, so it came from wild birds to geese. And then a year later, many people in Hong Kong got sick from this virus, and that began the current worry about it. But as you know, something is different now than then, and that's because in 2020, this B clade started to rapidly expand, first in Eurasia, and then in 2021-22, it crossed the Atlantic from Europe via Iceland in migratory birds. It had never done that before, okay? I don't know why it did it then. Maybe it got tired of Europe, but it then spread rapidly in the Americas and has even reached Antarctica. And here's a quiz for everyone. What is the only place on the planet? What is the only country that does not have H5N1 influenza virus? And you guys can answer it too. It's Australia.
Right, yes. Actually, they mentioned that in the paper, I think.
Yeah. So this now is well established. This clade B is well established in wild bird populations, except for Australia. And it's caused issues in domestic poultry. It has infected scavenging animals. There's mammal-to-mammal transmission in fur farms, marine mammals. There's an outbreak in dairy cattle. So that virus has gone into dairy cattle and has spread through the dairy industry.
This may be a unique instance of a bad introduced species that has not shown up in Australia.
Yeah, I'm very confused about the not Australia thing.
Well, you know, they are isolated from the flyways for the most part.
So these in creatures other than birds, it is not highly pathogenic, correct?
Well, in cats, it can make cats sick. It can kill cats.
I think it killed some of the marine mammals and some of the other mammals.
Yeah. Okay.
Yes, the marine mammals for sure it killed, and others, but not all. So
and is transmissible in some of these other species.
Yeah.
So this is something to keep an eye on.
Yeah, and it appears to be pretty transmissible in dairy cattle. Doesn't seem to kill them for the most part, but it has spread. We know of at least 70 human cases, 6 severe, 2 mortality, one in the U.S. and one in China. But most of the human cases have been mild.
All right, so now back up a little. After the 2010s, after this virus emerged in 2010 in Asia, this clade 2344H5 has reassorted with other avian influenza viruses to produce what are called H5NX viruses. So the X could be different neuraminidase, including N2, N5, N6, N8, or N9, right? However, this 2344B, which is the virus currently causing outbreaks, is an H5N1, okay? So it's not one of these other neuraminidase. Now, this has at least 2 things that are important for its pathogenicity. One is a polybasic cleavage site in the hemagglutinin. The hemagglutinin, which is one of the 2 spike proteins in the viral envelope, needs to be proteolytically cleaved to enable fusion and entry of the virus into the host cell. In most human influenza virus strains, this is done by a protease in the respiratory tract, and only in the respiratory tract. It's not present in other tissues, which is why most influenza viruses are restricted to the respiratory tract. However, H5N1 viruses have a different cleavage site in the HA. It's a furin cleavage site.
Oh my gosh.
Somebody must have put it there deliberately.
And furins are present in all tissues, and that's why H5 can multiply in many tissues and cause severe illnesses. Okay, so that's one of the virulence factors of H5N1. And also the neuraminidase is important, right? The neuraminidase helps, neuraminidase digests sialic acid from glycoprotein. So it helps the virus get through cut through mucus, cut through the mucus. And also on the way out of the cell, it can release itself from being grabbed by sialic acid receptors. So this activity of the neuraminidase, it's called, neuraminidase is the enzyme name, or receptor destroying enzyme. This is always balanced with the hemagglutinin, which binds sialic acid, right? So you can't have the neuraminidase more active than the HA, otherwise the virus would never get into cells. Okay.
Right, it would be cutting off its own receptor before it even got into the cell.
Cutting it off to spite its face, right?
Yeah.
Mm-hmm.
All right, so now the main subject, the stalk. The neuraminidase is a glycoprotein perpendicular to the viral membrane, as is the HA. And it's stuck in the membrane, and then there's a stalk, and then there's a globular head. And the stalk can be long or short, all right? The long ones can be shortened, and that has an impact on neuraminidase activity. And these truncations have been found in N1, N2, 3, 5, 6, and 7, mostly in gallinaceous hosts.
Yes.
What does that mean, Alan?
Foul.
I was going to say chickens.
Chickens. Yes.
Murder most gallinaceous.
Yes.
Yes.
But we don't understand these truncations, all right? They can affect glycosylation sites, right? And there's some evidence in mice, chickens, and ducks that truncations are associated with increased pathogenicity and in some cases increased transmissibility. But it's all different. In every species, it's slightly different. But there's one study which is cited here, and that's the basis for this paper being published, I think. One study, they increased the stalk length of neuraminidase and it increased ferret transmission. Okay? So that's why this paper says, okay, the current circulating H5N1 viruses have long stalks, so they could be more transmissible. But it's based on a ferret study, and I don't know what that means for people because ferrets are not humans. Did you know?
Yes.
Okay.
Some humans may resemble ferrets, but that's different.
What they did here is, now we get to the results. They wanted to see how this neuraminidase, this N1 neuraminidase has evolved starting from the 1996 lineage. They downloaded sequences and aligned them, and they got these from a lot of different species: chickens, ducks, turkey, teal. The original Guangdong 96 isolate had a long stalk, but then they saw 20 amino acid long truncations, which they call short stalks, appearing in the 1990s. And from 2000, this is interesting, from 2002 onward, the long stalk was mostly absent, and then it reappeared in 2022. Okay, so it probably didn't disappear. It was probably somewhere, and we didn't sample it, and then it just came back for whatever reason. All right, so then they said, let's look at more sequences. So they downloaded more sequences, and they find that the long stalk is the majority of sequenced H5N1 between 99 and 2001, but afterwards, the fraction with the long stalk declines, and then in 2021, it becomes dominant again. So there's this back and forth short, long stalk business going on here. Right now, 2344B, the currently circulating H5N1, have a long stalk neuraminidase. 100% of mammalian samples, 99% of avian samples, and these come from chicken, mute swans. Is this mute swan? Does it make any noise? Is that how that works?
I'm not sure.
It's an interesting name.
That sounds like it would not make any noise.
Yeah, I think those are the ones that are, speaking of invasive species, actually invasive in the Chesapeake Bay area now.
Foxes, cats, dairy cows, okay? They're all long stalk neuraminidase. And interestingly, they point out 65% and 80% of the viruses from wild bird and poultry species in 2024 have a long stalk, which is identical to the 96 Guangdong isolate. Identical. I mean, this is only 20 some odd amino acid longer, but still, you would think it's a long time ago, 1996, right? So they say this maybe in birds, it's evolving quicker. They also find some novel deletions in the database, which come mainly from West Africa, but they don't seem to be widespread or spreading. They have this nice picture here of the percentage of long stalk in clade 4B, and they have poultry, wild birds, canine, feline, cattle, and human. And it's like 98.5% or higher in all of them. 100% human, 100% cows, 100% cat, 100% dogs. And then the wild birds and poultry have a little bit less. Okay, so that's the stalk. Now, on top of the stalk is the head, which has the enzyme activity. The sialic acid goes into the head, and then it cuts it off, right? And so they say some sites in the head, which changed from 98 to 2019, seemingly reverted in 2020-21 to what was observed in 97. And they say if they analyze the evolution of these only in samples that have a long stalk, they say there's no head changes. So the viruses with long stalk have been present since at least 96, and they're somehow limiting the evolution of the head domain. I don't know what that means. But they say their conclusion is it's possible that the early spread of H5 in Eurasia was more driven by poultry-to-poultry transmission, where the short stalk version may have increased fitness, while current transmission may be driven by wild birds, where the long stalk has a fitness advantage. And so maybe that explains the change from short to long stalk dominance, okay? Chickens, think poultry, short stalks work better. Wild birds, long stalks work better. And so right now, lots of circulation in wild birds. So that's mostly what we see, long stalks.
So to me, what I'm thinking the whole time here is that the concern, well, there's lots of concerns, but a major concern with these H5N1 viruses is that they will acquire transmissibility, human-to-human transmissibility in humans. So the genetic characteristics that govern transmissibility are of significant interest. And so I'm looking at the data and the charts. They don't comment specifically on this, but it looks to me, or I can ask the question, in these sort of more recent observations of transmission, for example, in cows or other mammals, are those all associated with long stalk? It looks to me as if that's the case, right?
Yes.
It is, yeah.
Okay. So obviously, since it's not transmissible in humans at this point, that's not the whole story. Okay.
Exactly.
But it's
exactly.
part of the story, potentially.
Right.
Okay.
And
Maybe.
that in their previous work, where the previous paper they cited was the big part of transmissibility in ferrets.
Yeah.
Right.
I mean, it could be that you need a long stalk to then become transmissible with other changes.
Right.
So if that's true, then this is a bit worrisome.
Right.
Yeah.
But we don't know.
Right. And whenever we do something like this, my mind goes back to the Kawaoka and Ron Fouché days.
Yeah.
How long ago was that?
2015.
2015. And what
Not
I'm
so long
referring
ago.
to is the flu transmissibility and genetics experiments that set off the gain of function argument. Okay?
Yep.
Because when I see this kind of stuff, I want to go back to those papers and and all the papers since and catalog and try and understand what the genetic factors are that govern transmissibility and how that relates to species and everything else. It's extraordinarily complex. But,
It's a good point because I don't know if the virus they started with had a longer short stalk, right? And they were able to get it to transmit, but the stalk length didn't change.
was that an H5N1 virus?
Yeah, it was
Okay.
H5N1.
It was.
But the paper cited where they make the stalk longer and it increases transmissibility in ferrets. That maybe they worked with a short stalk variant in the Kawaoka Fouché experiments. I don't know. It's a good question, but I don't know what the ferret infections mean, frankly, because they're not people. But in those experiments, they were able to identify amino acid changes in the HA that gives rise to transmission in ferrets, but they'd also lost its pathogenicity. So
Right.
I don't know what any of that means.
Yeah, I mean, but to Rich's point, we have these data accumulating from studies like this, from the Fouché and Kawaoka, from everything's been done between and before. And there are definitely people working on this problem of what I think of as sort of correlates of pandemicity, if you will. How worried should we be? Which flags should we look for? And that's it's really a bioinformatics problem.
And I don't think you can know how worried we should be.
No,
okay,
no. Not yet.
What you have to do, and the next paper addresses some of this. What you have to do is that, I'm reminded of going sailing with my buddy Henry Baker, and we have an agreement when we're sailing with each other that this is sailing stuff, right? When the wind gets too blustery, when it gets too windy, you can get in trouble. And one way to deal with that is what's called reefing the sails, take in sail, all right? Which is much easier to do and much more difficult to do when it's too windy, okay? We have an agreement that the minute either of us think, I wonder if we should reef the sails, you vocalize that and you reef the sails. Okay?
Yes.
So if there's a virus out there, okay, that you're thinking, geez, I wonder, this infecting people, it's transmissible in cows, it's time to reef.
Yes.
Yes.
Okay?
So you want to make a vaccine.
Make a vaccine.
Right?
Exactly.
Make a vaccine, and study the heck out of that virus and figure out what countermeasures are going to be effective and be ready.
And I've actually got a pick of the week that speaks to this as well. So
And one thing I also would hope, I don't know enough to fully know this, but I wonder if all of those studies that we're mentioning here, where people have tried to look for these correlates of pandemicity, or whatever we want to call it, have gone past the bioinformatics stage and maybe actually tried to do things like use AlphaFold to look at structures, because maybe it's different sequences each time. But if we actually look at a 3-dimensional structure, we see, oh, it's always this part of the neuraminidase that is changing, or always this part of the HA or something like that.
Yeah. By the way, I am not worried about H5N1. I'm worried about our ability to make a vaccine against H5N1.
I'm actually, at this point, a little worried about our ability to make the seasonal flu vaccine this year.
Yeah, that too.
But, yeah.
All right, a few more points from this paper. Different clade, 2.3.2.1c, caused some severe cases in Cambodia recently, has a short stalk. Okay? Doesn't transmit among people, but caused some severe cases. And then recently there was an H5N1 infection in Missouri. We don't know the source, but it is a clade D virus, which is the avian clade, which is very pathogenic in people, unlike the B, the cow clade. All right. There's only infected one person, but the stalk of the neuraminidase is long for whatever that's worth. Okay. And so what they say is that, and in fact, this genotype has caused 2 recent severe infections in humans in North America with 1 death. It has a different N1 from North American avian influenza viruses through reassortment, although it has a long stalk. So the bovine clade is a different N1 than the avian strains that are circulating. Okay, so the conclusion of this paper is that the clade 4B viruses, which are currently circulating extensively, their neuraminidase has a long stalk, and that could lead to increased mammal-to-mammal transmission based on a previous paper done in ferrets, which I don't know what that means, but I liked the discussion of the H5 genetics. That's all.
Yeah. Yeah, and regardless of which specific changes are actually needed, which we don't know, the more chances you give the virus to spill over, the higher the probability that one of them is going to be a go.
Mm-hmm.
So we've got these dairy cattle that are infected all over the country, and we've got people whose job is to milk them, and it's just, this is going back and forth, back and forth. Who knows?
Yeah, so sort of in relation to some of the news articles, in both cases, you give viruses more rolls of the dice, whether those are among people who are not you or whether those are among cows, that could lead to risk for you. And so you should be aware of that, and you should be trying to prevent that.
Okay, our next paper, Nature Immunology, System Vaccinology Analysis of Predictors and Mechanisms of Antibody Response Durability to Multiple Vaccines in Humans. So durability means length, how long the antibody response lasts, okay? Not hard. If you hit it with a hammer, it won't break. Durability is the length of the best.
And
This is very cool.
Do authors and a little bit of an introduction here.
Right.
Brianne is going to be shouldering most of this one, I
So,
think.
you know what? The last time it was up to me to pick a paper for immune. This was on my short list. So
Okay, good,
I felt
good.
okay.
Good.
So the authors, we have 2 co-first authors, Mario Cortese and Thomas Hagen, and the senior author is Bali Pulendran. And they are from, damn it, Nature, why do you do this?
It's horrible.
Scrolling. And this is a long paper, so I got to scroll way down here. And you can't just go to the end, because the end is like after all the supplemental figures, but this is right after the footnotes. There we go. All right. These authors are from School of Medicine, Stanford University, Cincinnati Children's Hospital Medical Center, University of Cincinnati College of Medicine, Hope Clinic of the Emory Vaccine Center in Decatur, Georgia, University of California, San Diego, in La Jolla, California, whatever, GlaxoSmithKline in Rixensart, Belgium, Hospital Israelita Albert Einstein in Sao Paulo, Brazil, Jackson Laboratory for Genomic Medicine in Farmington, Connecticut, the Food and Drug Administration in Silver Spring, Maryland, the Icahn School of Medicine at Mount Sinai in New York, the Yerkes National Primate Research Center in Atlanta, Georgia, Emory University School of Medicine in Atlanta, National Institutes of Health in Bethesda, and NYU Grossman School of Medicine in New York. I could have just said these authors are from everywhere.
So,
and I just, to set this up, they have a great line here that the durability of vaccines is one of the great mysteries in immunology. Why is it that somebody who gets a measles vaccine when they're a kid is, pretty much immune for life, and somebody who gets a flu vaccine isn't even immune from the same strain of flu the following year? Why? Well, we're not supposed to ask why questions, but they're asking how can we look at that? And what they do is they round up 50. Yeah, so they get 50 volunteers, and they give them flu shots, either just plain influenza inactivated, it's an H5N1 strain, or that plus an adjuvant, which is an additional chemical in the vaccine. No, it doesn't cause anything bad. It's been tested extremely rigorously. People get with the program. Totally safe. But what an adjuvant does is it revs up the immune system. So there's a little extra thing in the vaccine. If your immune system didn't quite get the memo that this is supposed to be a threat, because you threw in a virus that's inactivated and the immune system's like, yeah, right. You throw in a little bit of a chemical that says, hey, this is for real, and then the immune system goes off. So these have been used in vaccines for 70 years. But they try plain versus adjuvanted vaccine, and then they do all the immunology on these volunteers and try and figure out what the differences in responses are, and then they do animal experiments, and it's a massive amount of work, and they have some conclusions about what the correlates might be of more durable immune responses than what we would see in some cases.
let me talk a little bit about adjuvants, because this is comparing adjuvanted and non-adjuvanted
Right.
H5N1 inactivated vaccine, as Alan said. So these have been around for 70 years. Most of that time, only insoluble aluminum salts were used. But in the past 30 years, more have been developed. And here's a list, in case you wanted to know. Oil and water emulsion-based adjuvants called MF59 and ASO3. These are in influenza vaccines. Then adjuvants containing the toll-like receptor 4 agonist, monophospholipid A. These include AS01 and AS04. Those are used in respiratory syncytial virus, herpes zoster, and human papillomavirus vaccines. Then an adjuvant called CpG 1018, which is a toll-like receptor 9 agonist. It's a CpG dinucleotide, oligonucleotide. It's used in the hepatitis B vaccine. And then finally, Matrix M. Enter the Matrix is in the recombinant COVID vaccine and the R21 malaria vaccine. It is a saponin-based adjuvant. So most of them, we don't know really how they work. Obviously, the toll-like receptor agonists are tickling the innate response.
Yes. So because I really like a lot of things that are discussed in this paper, including I really like adjuvants, I went down a rabbit hole about adjuvants a little bit. And I found a really nice, it's actually a book chapter that's reviews the history of adjuvants and gives all of the mechanisms of how every adjuvant we know about works. I just put the link in the show notes of this review that I found that I thought was fascinating because,
Cool.
Of course, I did. And they actually talk about the person who first just came up with the word adjuvant and how he defined it in 1924. And then everything we know about sort of mechanism of action of all sorts of adjuvants. And so they do certainly note all of the ways that we don't understand some of the details of how these adjuvants work, but at least for ASO3, they cite some data on it seeming to stress or damage cells so that they start making some inflammatory products as a result of that damage. But if someone wants to read about it, I got a lot out of this review.
So adjuvant is the Latin word, which means helping towards.
Mm-hmm.
All right. Okay, so the clinical trial, 50 people, 21 to 45 years. They get 2 doses, 21 days apart, of a monovalent inactivated H5N1 vaccine, which is from 2005. So I'm not sure how relevant it would be today, but it doesn't matter for this study. They get with or without the AS03 adjuvant, right? So it's oil and water-based emulsion. Adjuvant.
And it AS03 specifically contains, one of the things is alpha tocopherol,
Right.
which is vitamin E, and that is apparently a big part of the active ingredient.
I see. Then they do gene expression analysis of peripheral blood mononuclear cells, and they measure things that are important for B and T cell responses. So they want to know the impact of AS03 on gene expression in PBMCs. So they identify differentially expressed genes before and after vaccination. They're called DEGs, in the adjuvanted and unadjuvanted groups. And we find that AS03 gives you a stronger transcriptional response than without AS03. And the kinetics of the response in unadjuvanted recipients is also delayed. Okay, so what pathways are we talking about here? So the adjuvant increases. So we did this a similar paper a few weeks ago, where they use a computational approach using what are called blood transcriptional modules. Right, these are proteins that are turned on, and they use them to analyze human blood transcriptome. So they find that AS03 increases the expression of modules associated with the broad range of innate and adaptive immune cells and pathways. In particular, they find these modules related to monocyte and dendritic cell activation, enhanced in the adjuvanted group, but in the people who didn't get adjuvanted, very little immune activation. And even when they got a dose, it was still not as good as in the AS03 group.
Yeah, it definitely was noteworthy how much earlier the A503 group started making a response.
I would like to have adjuvant in my next vaccine based on just that response alone. Okay, so they noticed some differential transcriptional activity after the adjuvant is dosed. In particular, some genes related to interferon signaling and activation of dendritic cells are more strongly upregulated, and also genes encoding proteins with chemotactic functions for granulocytes, monocytes, and macrophages. So they think that basically the adjuvant increases these specific innate responses longer following the booster immunization. And they say that this is reminiscent of other studies. They had done some previous studies on COVID vaccines and influenza vaccines, and this is reminiscent of that. Now, one of the predictions is that the adjuvant should enhance antibody responses. So they look at this specifically, and in fact, the adjuvant increases the neutralization titers at every time point measured following adjuvant vaccination versus no adjuvant at all. And the unadjuvanted, they have much lower neutralization and hemagglutination inhibition responses. So furthermore, these antibodies are cross-clade. So we were just talking about clades, right, of H5N1, like the cow clade and the bird clade. These antibodies will neutralize across clades. So somehow the adjuvant is doing this. And they say it's probably inducing antibodies against the head of HA, which is less conserved across clades.
By the way, just because it kind of slips under the radar in all the weeds here, we're doing clinical trials on an H5N1 vaccine, okay?
Yes.
Mm-hmm.
Which is what we were just talking about as a potential threat to humanity, okay? And trying to understand how to make the best vaccine we can in a global collaboration using NIH dollars, among other things.
really global,
Really
Yeah.
global.
as it should be. Science is always crossing political boundaries until the politicians get involved. They looked at the affinity of these antibodies by a wonderful technique called surface plasmon resonance. And they find if you get the adjuvanted vaccine, you get higher levels of HA binding antibodies to both the head and the stem after the prime and the boost. Okay, so let's talk about what's going on here. What is it doing? So they say these high affinity antibodies, maybe T follicular helper cells are involved. These are involved in affinity maturation of antibodies in germinal centers to get higher affinity. So maybe these are playing some role here. So let's look at that. And now, of course, these people gave blood, right? Mm-hmm. So they look in the blood and they find some cells circulating that look like T follicular helper cells. Now, that's not normally thought to be in the blood, right, Brianne?
Yeah, they usually are in the germinal center in the follicle. But fortunately, there are sort of these related cells in the blood. But the fact that I'm so glad that you mentioned the blood, because that becomes such a big part of this later.
Yeah. So, in fact, the presence of these TFH-like cells in the blood correlates with higher neutralization titers of the antibodies. So there's a correlation there that could be interesting.
Yeah.
So
I will say at this point of the paper, before they go into their next step, I'm sort of like, yep. A lot of this is very similar to some previous studies, and is showing things that maybe I might not have, or I might have predicted. I might not be super surprised by. They look at, a lot of this really looks at things that have been done in other studies, but then when they go to this next part, it's all things I hadn't seen before that seemed really novel.
So they did some experiments to characterize these blood cells in terms of the markers they have, and I don't want to go into that, but I want to summarize before we get into the cells that are involved here. So the adjuvanted vaccination, you get higher frequencies of circulating activated T follicular helper cells, and these frequencies correlate with neutralizing antibodies and affinity of these antibodies to the head of the H5. So somehow ASO3 is driving T-cell dependent mechanisms of B-cell affinity maturation and antibody production, right? The T cells are the TFH-like cells. All right. So back to the original question. How do you make long-lasting responses to vaccines? And so they take it's what they call a day 7 plasmablast signature, which they've seen. So plasmablasts are the precursors to the cells that make antibodies, right? The plasmacytoid. What are they called?
Yeah. So the plasma cells are the ones that are making antibodies.
Plasma cells. Yeah.
And the plasma blasts they're talking about are sort of the cells that are getting ready to become those plasma cells.
Right.
But I think that the really cool part here is that they said, okay, we've always looked to see what is going on at sort of that peak time after we give the vaccine at maybe a short number of days, and what if you look longer, and specifically, what if we try to figure out, is there something we can measure at an early time point that predicts how well your response will last, how well that will persist, which they refer to as durability. One of the first things I looked for when I was looking at this experiment was what they meant by durability. So most of what they're looking at is looking at day 100 following vaccine, which is pretty good. Most people don't look that long, though, some people might argue, well, I want to know if my vaccine is going to give me good antibodies for 5 years or something. And so if anyone's wondering, the durability here is 100 days. It goes out a little further in some of the other experiments, but that's what we mean with durability.
Okay.
And they just want to know what early time point markers correlate with having a good response later.
And a major reason you want that information is because the longer the trial, the more expensive it is, and the more people you lose to follow up. So yeah, they followed for 100 days. That's a lot. And we'd
Mhm.
love to know what happens 5 years down the road, but good luck finding
Yeah.
all your volunteers in 5 years, and who's going to even fund your study for a 5 year. If you're trying to develop vaccines, what you'd like is a test you can do, within a couple of days. So you've got your volunteers, you bring them back in, you test, and you want to be able to predict, oh, that looks like the durable response, that looks like a less durable response. And so this is for future studies that would be structured that way. They want to find what those early markers are.
Yeah. And they knew that previously having a good plasmablast signature
Right.
at day 7 correlated with high titers of a response at the peak. And so they'd always, their goal in the past had always been, we want a vaccine that gives you good day 7 plasmablast signature.
Yeah.
Um,
However, but
yeah.
it didn't correlate with durability, though.
Yeah, and so now they're like, oh, wait, it doesn't actually
This
correlate
isn't
with
good.
durability. It correlates with peak, but not durability.
So then they say, okay, let's see what correlates with durability. So they do gene analysis of their sequence data, and they look at genes ranked by their correlation with day 100, day 42 antibody titers, and they find that genes within the platelet activation actin binding module, a particular module containing a bunch of genes, showed strong agreement with antibody persistence. And so this is in this human cohort, right? So then they do an experiment in rhesus macaques.
I got to say, when I got to this point of the paper, I had not seen platelets coming.
Yeah,
No, I didn't.
nobody saw platelets coming.
No, I thought, what the hell? What the
Signatures
hell?
in
Platelets,
what?
come on.
What the heck are they doing in there?
Yeah,
And
and
if-
in fact, I ended up on another rabbit hole with this paper because they did a lot of their work in PBMCs and they were looking at transcription in PBMCs, and I wanted to know about levels of platelets in PBMCs and levels. They'll later talk about megakaryocytes, which make
Right.
platelets, and levels of megakaryocytes in PBMCs to know who was actually in the sample that they looked at. So
So,
there are platelets.
Can we take a little time out here and have Brienne explain to the listeners what platelets are and where they come from and why we're surprised, maybe?
I can try. I don't know that
Give it
I
a
am
rip.
enough of a platelet expert to fully get this. But platelets are actually cell fragments. There's originally a cell type called a megakaryocyte that is in the bone marrow that results in the production of these little fragments of platelets that are really important in blood clotting. And so usually we think of, I honestly, when I do a lot of experiments or when I've done experiments with PBMCs in the past, I think of platelets as like icky contaminating stuff. Not really something that we think of as being particularly immunologically active. And we also usually are thinking about them as cell fragments. And so the idea of where they later talk about, genes expressed in platelets, I was like, wait a minute.
Yeah, right. How can that be?
Yeah, and
and
so-
platelets.
there's no
right. So
nucleus.
in the process of making platelets, as Brienne said, these aren't cells really; they're bits of
Pieces.
cells. And I always, just from the name, I always think of clotting as this process of throwing plates that are going to shatter and make the clot, and that's what's happening. The job of the platelets is to just be shrapnel. They're not even cannon fodder. They're a tool, and they don't have DNA. They have RNA that was given to them by the megakaryocyte, and that RNA is all they have to translate their basic proteins with. And as that RNA breaks down, the platelet just falls apart, which is its job anyway. So yeah.
So correct me if I'm wrong, but since they don't have a nucleus, they're basically pre-programmed to do anything that they're going to do. Well, it doesn't mean that they can only do one thing, okay? Because you could have you can have pathways go off. And the way I think of them is that they circulate and they can home to sites of injury, okay? And when they find a site of injury, they stick there. That's where adhesion molecules come in, right? At least in part.
Partially, yeah.
And they actually sit down and change shape and plug the hole. and also send off other signals saying, hey, I've got my finger in the dike here, but I need help.
Yeah. And to be honest with you, that description is an awesome description and is in fact more generous than I think some immunologists would be to platelets in terms of the how much activity or how much that they're actually doing. Because that was sort of, to me, one of the just mind-blowing parts of this paper was like, wait, the platelets are important in doing things?
Yeah. I mean, they're basically thought of as sandbags.
Sandbags, that's good. I like
You know, block it up.
Sandbagging. So they validate. So what we have here is we have a platelet transcriptional signature from day 7 post-boost, which correlates with day 180 antibody levels. So they validate that in a rhesus macaque model. And you may think, well, what's the importance? Well, the importance is you're going to be testing vaccines in rhesus macaques. So you can use this to say, is our formulation giving longevity without having to wait 180 days? Right? You could look at the platelet transcriptional signature. Then they use machine learning to say, is this signature going to work for other influenza vaccines? They have a lot of data from other studies of influenza vaccine responses. So they feed the machine learning program with all the data that they have, and they asked it to build a model where you predict the antibody level based on these transcriptional signatures. And in fact, they can look at multiple vaccine studies, and this works according to this model that they have built. The genes are platelet-derived genes that we've just been talking about, which are correlating with antibody persistence. Okay, and in fact, there are a bunch of specific genes that they mention that include cellular adhesion and migration genes, which are highly expressed in platelets, and they say these. So this is a potential role for migration in the establishment of durable immunity. Okay, so what is the cellular origin of this transcriptional signature? So they do a certain type of sequencing and construct what they call the transcriptome landscape of PBMCs on day 21 and day 28 from 3 people who had durable antibody responses and 3 that had falling antibody responses. And once again, the antibody longevity-related genes in this signature were highly expressed in a cluster of genes that are platelet-derived, and also in some genes that are both in platelets and monocytes. Right. So they go through. So basically, most of these genes are expressed in platelets, but they're also in other cell types, including monocytes, B and T cells, NK and DC. So it may not be as simple. Mechanistically, it's not as simple as platelets, but you may be able to just use platelets as a predictor.
Yeah, I mean, they also here really helpfully, we're thinking about this idea that, well, maybe we're getting this weird signature because like platelets are stuck to stuff.
Yeah,
and so
right.
we're not actually getting platelets because platelets, what?
Yes.
Yeah, let's make sure we have that technical distinction, because most of the experiments are being done with PBMCs, right? For peripheral blood mononuclear cells. So that's just a fraction of the total blood volume that has been purified to enrich for the immune cells, right?
Right. And in theory, it's not supposed to contain platelets, but it always has lots of contaminating platelets. And some of them are stuck to other cells because they're sticky. And so they did do the controls to show, no, these are really platelet transcription things. It's not that we're just seeing platelets stuck to stuff, and it's really this weird combo of a platelet and a monocyte stuck together that we're getting these transcripts from. By using single cell sequencing, they can show that no, it's actually the platelets.
Yeah, and in their model they can leave a gene out and like a gene set from PBMC and show that they don't really help. It's a platelet gene set that's really contributing to the longevity. So they did some further analysis, which is really interesting. So there are genes that are correlated with longevity and antibody responses. They have differences among plasmablasts in spliceosome and electron transport expression. And they find that plasmablasts of persistent responders had elevated expression of genes encoding small nuclear ribonuclear proteins, which are involved in splicing, and mitochondrial respiratory electron transport complexes. And they say this is consistent with findings of others, which indicate that oxidative phosphorylation and mitochondrial remodeling are needed for B cell activation and antibody production by plasmablasts. So you're identifying in your signature the genes that are needed to make antibodies. It's very cool. Okay, so platelets are made from megakaryocytes, as we learned. And they say, okay, well, the megakaryocyte should have a similar transcriptional signature, right, as the platelet. And maybe that affects, so maybe antibody durability is really a reflection of megakaryocyte activity in the bone marrow.
And just to continue on this theme, megakaryocytes are in the bone marrow. They're not going to be in the peripheral blood, right?
Right, exactly.
But they're the source of platelets.
Right. So there are some factors that are produced by megakaryocytes, including APRIL and IL-6, that are plasma cell survivor factors, and TPO. What is TPO, Brianne? What is this?
Thrombopoietin.
Thrombopoietin increases, which is made by, if you activate megakaryocytes with TPO, they can increase survival of plasma cells in the bone marrow, those long-lived plasma cells. So they do an experiment where they give mice TPO, and those mice make higher antibodies after they get immunized with spike protein of SARS-CoV-2.
And this is also a really important thing to think about, because you might say, oh, if it's the megakaryocytes, that's great, but we're probably never getting bone marrow samples from large numbers of people to do megakaryocyte testing. So the fact that you can actually look in the blood for megakaryocyte produced factors is important.
Yes. That's so funny they said that. We don't normally get bone marrow in a vaccine clinical trial. Like tongue-in-cheek. So this TPO effect, they have knockout mice that lack the receptor for TPO, and these mice actually have lower numbers of megakaryocytes, and they have reduced total serum levels of total IgA and IgG, as well as bone marrow plasma cells. So there seems to be a role for megakaryocytes and TPO in the survival of plasma cells and antibody production. And if they give these mice TPO, the knockout TPO receptor mice, they don't increase their antibody after vaccination with SARS-CoV-2 spike program. So somehow TPO signaling is promoting antibody response durability. So they do an experiment where they culture in vitro murine plasmablasts with TPO-treated megakaryocytes from the murine bone marrow. This increases the plasmablast survival and IgM antibody production compared to cells that don't get TPO. And physical separation of the megakaryocytes and plasmablasts, they put a membrane in between them, doesn't work. It doesn't increase antibody production. They have to be touching each other in order for this to work. So that's very cool.
I think this in and of itself is something that a lot of things could come from, because we make antibodies in a dish. We make antibodies in vitro in a lot of situations. And so I wonder, in any of those in vitro antibody production situations, has anyone ever tried to add some megakaryocytes or some TPO treated megakaryocytes, and would that increase your antibody production?
I bet somebody's tried it now.
Yeah.
So they repeat this experiment with human bone marrow, megakaryocytes from human, and plasma cells from human bone marrow. They treat it with TPO and look at plasma cell survival and antibody production. They get the same results as they did in mice. So basically, TPO activates megakaryocytes. It boosts antibody production and durability to vaccines, probably by promoting survival of plasma cells in the bone marrow via these factors, APRIL and IL-6 and so forth. And it depends on contact, which involves a specific series of protein-protein interactions. How about other vaccines? Can this platelet transcriptional signature predict longevity of other vaccines? So there are lots of databases they could tap in order to do this. They looked at yellow fever vaccine, meningococcal vaccines, malaria vaccines, COVID mRNA vaccines. All of them induce an antibody response that goes down, except yellow fever vaccine, and that correlates with the platelet transcriptional module. And yellow fever is just really good, and it would last 3,000 years in people, so they can't even apply their model to that. Let's see, what else? They make a model to test this study I just told you was using actual data, transcriptional data from these vaccine studies, so they could look for their platelet transcriptional group. But now they make a model, a computer model, to say, can we predict antibody persistence to other vaccines? And they say it can do it for most vaccines, with the exception of yellow fever, which is off the charts. And many of the genes are in these platelet signatures, but some of them are in other cells as well, and that means that there are other mechanisms involved, not just the megakaryocyte-dependent ones. So that's the cool result. So you get this transcriptional signature from platelets, which correlates with longevity of antibody production. So this platelet signature is a primary driver of the longevity of vaccine antibody responses to vaccines. It's really remarkable.
And mechanistically, just to review for my own sake, this is correlated or maybe a function of interaction between the platelets and the plasma cells that increase their, or do we know that much?
I don't think we know that much.
Their experiment suggested it's the interaction between megakaryocytes and plasmablasts induces long-lived plasma cells.
I mean, right? I think it's somehow that the megakaryocyte is making platelets, whether it's making better platelets, different platelets, more platelets. Somehow the type of platelet that is made when the megakaryocyte gets TPO is a good kind of platelet. And that good kind of platelet is correlated with antibody production by the plasma cell.
Okay. But correlated with, and mechanistically, we're clueless.
And probably needs to touch it.
Yeah, they need to touch
it. Fine.
The megakaryocytes need to touch the plasmablasts. Right?
I was just going down my own little rabbit hole here and looking up platelet disorders. And wondering if, probably nobody's ever looked, whether there's any sort of correlation between platelet disorders and immune function, right?
Yeah, I don't know. I am sort of like, are there ways that we can modify, TPO production or involve TPO in vaccine adjuvants to influence this? And of course, does this change longevity of other parts of the immune response, like the T cell response?
They comment on the yellow fever vaccine, it induces a really long-lived antibody response, which doesn't vary among people. So you can't use their models to predict genes associated with decay.
What's your control? Yeah.
Yeah. It never goes down, right? It's amazing. Wow. So obviously you could design vaccines and then see if this signature is present. And if not, you go back and tweak them. I don't know how, with adjuvants or whatever. Well, they say, we need to do a lot more research before we can start doing that.
Figure out what induces this signature.
Yeah. In different populations too, also, right? It's maybe variable. But yeah, you can see that this could be very useful in the future.
Yeah.
And they say this is, what is it called? Systems vaccinology. So you've heard of systems biology. Now you got systems vaccinology. It's cool. All right. It took a little longer than I thought. So we will skip letters and go directly to picks of the week. But there is hope in science. As you see, it's amazing what science can
Yes.
do.
Yeah.
Don't shut it off.
It keeps going on. You can't shut it off.
Yes.
Brianne. I said Dixon. Oh my God.
Dixon's right
Yeah,
behind
Dixon's
Rich.
right here
It's right
in
there.
spades.
Brianne,
Yeah.
what do you have for us?
So I am continuing with the fact that many of us enjoy space-related things, and I know that earlier this year, I was so excited to be able to see the Northern Lights in New Jersey, albeit poorly. And I came across this video that is taken by some of the astronauts on the ISS as the ISS was going around the Earth. And they actually were able to take video of the Northern Lights from space. And so this is a short video showing what this video that the astronauts took from the ISS. So if you were excited about seeing the Northern Lights before, you've never seen them like this before. It's really cool.
It is cool.
Yeah. It is very, very cool.
Wow, that's neat. Rich, what do you have for us?
So in the realm of pandemic preparedness, etc., as I've spoken of many times before, I volunteer for this organization, Brighton Collaboration, and I get really cool feeds from them. And this is a bulletin, a news bulletin from the World Health Organization, says that titled "Who and Partners Enable Access to Candidate Vaccine and Treatments for Outbreak of Sudan Ebola virus disease in Uganda." I think we've mentioned this outbreak, have we not?
Yeah.
Yeah.
And the deal here is that they were able, the minute there was a case, the outbreak was basically declared as a single case of this, okay? And the minute this came down, they were poised with a candidate vaccine and were able to immediately start ring vaccination trials. The vaccine is essentially, it's the same platform that was used for the Ebola Zaire vaccination campaign, which is a vesicular stomatitis virus backbone with the Ebola virus glycoprotein in it. But this is the Sudan Ebola virus glycoprotein, which doesn't cross-react with the Zaire guy. But they knew Sudan was out there on the horizon, so they made the vaccine. They have a small stockpile of it so that when this hit, they were able to deploy it right away and start looking at its efficacy in a ring vaccination campaign. So this is pandemic preparedness the way it ought to work.
Sounds like something you shouldn't turn off.
No, don't want to turn that off.
Glad they were able to do this.
And my guess is, that we have much more robust stockpiles of various H5N1 vaccines. We just did a paper that riffs on trials for H5N1.
Yeah.
And there are organizations like CEPI and IAVI that are in the business of trying to identify pathogens that are of pandemic potential and get us up through preclinical trials and phase 1 trials to have enough vaccine there so that if something actually does start to look nasty, they can be ready.
So WHO donated $1 million from its contingency fund for this effort, right? That's why you need to pay your dues every year for WHO, so they can help other countries.
Yeah.
It's not all about you.
Yeah, but even if you think it's all about you, this still matters to you, because if you don't control it there, it's coming here.
Yeah, Alan, that's too much to comprehend.
Yeah.
Nobody's safe till everybody's safe, right? That's Daniel's
That's it.
right?
That's it.
That's it. Alan, what do you have for us?
So this is, I've got a news article in Science, and then I'm also including a link to the unfortunately paywalled paper in Current Biology. This is a cool little microscopy and entomology analysis. There are lots and lots of insects with camouflage, and there's this moth that's in this study that looks like a leaf. and it really, really nails it. It's not just that it's got the brown coloration and the shape and the the veins of the moth wing look like veins of a leaf. It actually manages to get the specular highlights of a leaf. Like if you were painting one, you'd get some of the white paint and you'd do some little, here's where the sunlight is glinting off, where the shape of the leaf is. So it's a whole 3D effect. And you can't build a wing like that because the 3D shape of the leaf would disturb the airflow. So the wings got to be flat, but how do you make it look 3D? And this moth has evolved specialized wing scales. So the wing is covered in these microscopic scales, and there are specialized scales that are just to do these specular highlights.
Very cool.
To get the 3D look. It's really, really cool.
It's nice.
That's very cool.
So if you can do that, you don't get eaten, right?
Exactly. I mean, that's the prize. It is the most brutal art competition ever.
Unless they like leaves, then you get eaten.
Yeah, but it looks like a dead leaf. It's not even an herbivore.
Yeah, it's brown.
It's brown.
That's right. Right. That's pretty cool. All right. So I have a post from Avian Flu Diary, which is a blog that's been going around for years. I remember when I started blogging years ago, 2006, this was going. And so this is by Michael. Where did his name go? Michael Costin.
Yeah.
Costin. WHO recommendations for flu vaccine composition for the 2025-26 Northern Hemisphere influenza season. So the WHO has a committee that meets and decides on the composition of the vaccine for both egg and cell-based. So you can see what they recommend here with the different strains. They did make some changes in the vaccines. And then normally they say, now the reason I like this article is that they say normally within a week, the FDA advisory group meets to recommend the formulation, but that's been postponed, as you know. And so it's not clear, how long it's going to be delayed because, as Alan said earlier, you have to start manufacturing this stuff now. And it's ridiculous.
If I were an executive at a vaccine manufacturing company, I would be PO'd right now.
Well,
This is a big problem.
Yeah, it's ridiculous. There's just no reason for it.
Yeah.
I mean, if it's really to find a bigger room, give me a break.
No, it's not to find a bigger room. It's to cause chaos. It's to yeah. That's the only motive.
It's just illogical and incomprehensible to me. The way I was trained as a scientist, this makes zero sense, and I don't know why anyone would do it. As I said, it doesn't make any sense.
Nope.
We have 2 listener picks. One is from Jerry. I was listening to TWiV 1195 this weekend. Much love to Dixon. When I heard the snippet covered different ways DNA could be translated to proteins, DNA, mRNA, proteins. Okay. I was reminded of a video which was recently released by a science communicator. It illustrates the process of DNA being transcribed into mRNA, including how different apparatus play into the action. It also promises several future videos that dig into how the processes work. Thought I would share it. Seems like a great high-level illustration of the topics which educators might find useful and which folks who listen to TWiV would appreciate as a primer on protein synthesis. So Jerry provides a link to a cool YouTube video of this process. Lots of great animations like this.
Yeah, I actually subscribe to this because it looks like it's going to be a whole thing, right? There's a precursor to this that's DNA replication. This is transcription. Protein synthesis will be coming up.
Nice.
Great. Transcription is cool. All right, we have one from Ann. Years ago, I took this course with Mike Brown of Caltech. Dr. Brown is known not only for helping to demote Pluto from its planetary designation, but also for the discovery of other large items in that region of the solar system that eventually led to Pluto's demotion. It was a fascinating course, and Dr. Brown is an excellent teacher. I don't know how much the course has been updated, but it will likely have more current information regarding the Juno mission. So links to the science of the solar system, an online introduction to planetary science
Cool.
at Caltech.
Nice.
I have to say, though, what they did to Pluto was pretty low.
I think it was a low blow.
That's really rough.
I do.
Maybe not as bad as what we did to Uranus, but
Yeah. So this class is serious, he writes, which is not the same as hard. It requires effort, but is not above the head of high school and above level student who puts in the effort. I like that.
This actually looks 9 weeks here. Bunch of
put in
different
effort.
lectures.
That's the way it works. If you want to be good at something, you need to put in the effort. If you want to be head of HHS, you need to put in the effort. I really believe that. You have to study, you have to read, you have to see what's going on, you have to consult people. It takes work. Don't be lazy.
In my experience, most people who are really good at stuff are that way because they're absolutely passionate about it. They can't not do it, okay? I've read several biographies of musicians that do this for me. Eric Clapton, Bruce Springsteen, right? They just couldn't not do it.
Yeah. So if you, you want to be a good teacher, you have to work at it, but also you have to have a passion for it. You can't.
You're thinking about it all the time. How am I going to explain this?
Yes, you can't do it because it's a job and this is how I get paid. But it's not good enough.
I just finished a book that has been on my reading list for years and years. I finally got around to it. Stephen King's book on writing.
Ooh.
Yeah.
Yep.
Which I highly recommend to anybody. But, it's really, really good. But a lot of it is just the fact that he can't not do this.
Right. So same theme. Which, to me, I just can't fathom at all. There's nothing I would rather not do than write.
Yes.
Yes, but Rich, when you had a lab, you could not go to the lab. You loved it.
Oh yeah. I looked, this is one of the reasons I think about this. If I had to do it all over again, despite some of the major pains in the ass, I would do the same thing because I couldn't not do it.
Yeah.
Yeah.
I loved working in the lab. I loved thinking about the problems.
I'm sitting here playing around looking at this website for this course because I really like space.
You don't have time to, Brianne.
I know. I know. And I'm looking at the syllabus and thinking about what you all said about Pluto. And as you were saying that, I was going to make a comment about Planet 9, but decided not to get too deep into my rabbit hole and how much I like space. But he has Planet 9 on his syllabus. He says, and we'll talk about Planet 9 because Planet 9 is real, people.
So, Brianne, do you want to go for a trip in space?
Sure.
So
Even after reading City on Mars?
Yeah, haven't finished all of City.
Oh, okay.
If someone said, Brianne, we'd like to put you on the mission to Mars, would you go?
I would definitely consider it.
Even if they said you may not come back to Earth?
Maybe.
I'd want to know who was going along with me.
Yeah, I'd want to know who was going along with me and if I had Wi-Fi.
That's what Kate Rubin. That's what Kate Rubins used to say.
That's
right?
who's going along with me.
Depends on it. Yeah. No, I'm out. After reading City on Mars, count me out for the Mars
I'm not
colony.
going to space. Nope.
Not doing it.
So I'm not City on Mars. Tell me about it. What?
Oh, that was one of my picks a few months ago. It's who are the?
It was really great, but then I ran out of time.
Yeah, that could happen. So it is Kelly and Zach Weinersmith, called A City on Mars. The subtitle is "Can We Settle Space? Should We Settle Space? And Have We Really Thought This Through?"
Okay, so this is nonfiction.
This is nonfiction. It is a fascinating read. It will go against a lot of what you've heard about, how we're going to have cities on Mars. And it's just a lot of fun too, because she is a biologist and he's a cartoonist.
Okay.
And there are cartoons that he drew that are in the book, and I don't know who did most of the writing, but whoever did the writing is also hilarious. It's a really, really fun read.
Okay, cool.
Go for it, Rich. All right, that is TWiV 1197. Show notes, microbe.tv/TWiV. You can send us questions, comments, pics, TWiV@microbe.tv. And if you enjoy these programs, we'd love your financial support to continue them. Microbe.tv/contribute. Brianne Barker is at Drew University. BioProf Barker on Blue Sky. Thanks, Brianne.
Thanks. I learned a lot.
Alan Dove is at alandove.com, turbidplaque.com. Thank you, Alan.
Thank you. It's always a pleasure.
Rich Condit is an emeritus professor, University of Florida, Gainesville. He's currently in Austin, Texas. Thank you, Rich.
Thanks, everybody. Always a good time.
I'm Vincent Racaniello. You can find me at microbe.tv. I'd like to thank the American Society for Virology and the American Society for Microbiology for their support of TWiV, Ronald Jenkins for the music, and Jolene Ramsey for the timestamps. You've been listening to This Week in Virology. Thanks for joining us. We'll be back next week. Another TWiV is viral.